feedback for the practical that you did in week 14. And as a reminder, you did the DNA isolation from banana. You also carried out what is called a mini prep of an E. coli plasmid, and you did precipitation of DNA with some fish DNA. And the last task, that was the agarose gel, and the demonstrators did this actually for you. So this is what we looked at when we prepared the agarose gel, when we loaded some samples. So first of all, here is an image of the agarose gel. It's in black and white actually because of the way we run the gel and we visualize the bands. We notice that at the top here we've got the wells where we actually loaded the samples. So here were our DNA samples that we put in right at the beginning. And the gel had a positive electrode here and a negative electrode up here, and we let the DNA run for about 1 hour or something like that. So here are the wells, and we loaded a marker with known fragment sizes. So the fragment sizes are here in base pairs, and I abbreviate that as base pairs. So here would be roughly 3,000 base pairs. Here this slightly stronger band is about 2,000 base pairs, and this relatively bright band here is about 1,500 base pairs. And we can use this marker to find out how large some bands are. Don't forget, when we plot DNA, we would use the logarithm of the size versus the distance that has been traveled. For example, this distance here in centimeters. So that is our marker. Now let's start with the banana DNA that we loaded onto the gel. Unfortunately, we didn't get a lot of DNA from the preparation. One of the problems that we had was probably that the cells were not lysed enough. And if the cells are not lysed enough, then they would not release a lot of DNA. But we saw a little bit of DNA. Now this is genomic DNA here. So genomic DNA. And as such, it consists of very, very large DNA fragments. And these DNA fragments actually don't really enter the gel. They are too big to go through the holes in this agarose gel. So they usually then will sit right at the top of the gel. They will stay in the well, which is absolutely okay. This is what we would expect for very, very large fragments. And don't forget, in agarose, we have these agarose strands that form these different pores. So this agarose. And the DNA would go through these pores, but if the DNA strands are too big, they will not enter the gel. So that's absolutely fine. Now we put a little bit of the plasmid that you isolated from the mini prep onto a gel, and what we find here are two bands. And as I pointed out in the lecture, the lower band, that is the supercoil DNA. And as a reminder, we have a plasmid DNA which is circular, and when it is supercoiled, it is really twisted up into a very compact structure. So that's the supercoil. And it's really quite small, and it can swish through these holes quite easily. Now the supercoil here, we would probably say that this is around 2,500 base pairs, but this calculation doesn't work because the supercoil is so small and so condensed that making any judgment about the size doesn't really make a lot of sense. In fact, the plasmid, so I was told, is about 2,000, probably 2,700 base pairs large. But the supercoil shows something smaller. The upper band here, this is the open circle. That's the open circle band here that we see, and that's absolutely fine that we see another band. This is actually when the supercoil has been relaxed, probably through a single sort of break in one of the DNA strands, because then it is a sort of a circular molecule which runs higher than the supercoil. And if we had linearized this molecule, this plasmid, then we would have seen a third band between the open circle and the supercoil. So what we are seeing here for the plasmid, that's exactly what we would have expected. And that looked quite good. There seems to be quite a lot of plasmid DNA being present. Now, with the experiment with the fish DNA, remember you carried out the experiment where you had the fish DNA. Fish DNA, you added some salt, plus salt, and you also had a sample without salt, and you added ethanol to these two samples. You also had a sample where you did not add any salt and you did not any ethanol. That was the sample that gives us the total DNA. Total DNA that you actually have used in your experiment. Now, in these two experiments here, you did a precipitation and you looked at the pellet. So in theory, you should have seen a pellet in this case because you had the salt and the ethanol. And in this case, where you did exactly the same but without the salt, you should not have seen a pellet because you could not precipitate the DNA without salt. And remember, the salt is important to neutralize the negative charges on the DNA through the sodium ion, the positively charged sodium ions that you add. And only when you neutralize these charges on the DNA, you can precipitate it. And the idea would have been, or the expectation would have been, that you see above a roughly similar concentration to the total DNA. You might lose a little bit through the precipitation, but you should get roughly the same absorbance. So if you've got an absorbance of 0.7 here, you would expect to see an absorbance of 0.6 when you resuspend the pellet, whereas in this case here, you should have seen a much lower absorbance because there is no pellet. But a lot of people actually said they didn't see a pellet and they did not see an appreciable absorbance here, which means that the DNA has been lost in the precipitation. So we wanted to see what's happening with this fish DNA. And we would have expected the fish DNA to be like the banana DNA. It is genomic DNA, so we would have expected the DNA to be up here. However, we see a very strong band down here. And this is a very small size, so we are probably in the range of, say, 50 to 100 base pairs that we see here. And very clearly, this is not what we expected, but it can be explained if the fish DNA, if this DNA has been degraded by some enzymes that destroy DNA, we would see exactly this picture. The genomic DNA totally degraded and broken down into very small fragments. And these small fragments actually don't pellet very easily. They don't precipitate very easily. And that is the reason why many people did not see a pellet. And when they measured it, they didn't see any absorbance. So that is basically the result from the agarose gel from this task 4. I hope it makes sense, and thank you very much for watching.